Krishnamurthy, who would talk about the multi-scale approach to study the biophysics of parasites and plankton. And here there is this PhD defense committee, including Professor Sanjeeva Lilly, Professor Bowen, Professor Mark Denny, myself, and needless to say, Professor Manu Prakash, bioengineering, who's been the primary advisors and host Deepak in all these years. So I pass the baton right away to Manu for an introduction of just maybe want to mention for the audience that this first public section is going to last roughly 1 hour, and then we're going to have a 10 minutes break, and then we're going to have a closed door session with Deepak and and the committee for the final part of the oral defense. And so, Manu, the stage is yours.
I'm absolutely delighted to welcome you all. I see many familiar faces. It is sad that we're not doing this defense in person, but on the other hand, I see people connected from around the world. So frankly, maybe all defenses should have a global component. And I see many familiar faces. Thank you all for coming. This is a very special moment. I want to take a few minutes just describing the individual that you're about to hear from. So you get a multicultural and a multifaceted view into the life of an individual. It's PhD is a long-term partnership and a collaboration, and I'm just so joyful to say that I'm so proud of this very day, and especially what Deepak has achieved in the lab over the last couple of years. So beginning from the left, I often enough, when Deepak is photographed, he's often seen smiling, and that tradition always continues. Many of you might not know this about Deepak, that he's also a fairly accomplished musician. I have not had as yet the chance to hear him perform, but maybe if somebody wants to ask a tough question, that might be the way to get him to actually perform. The other thing about this is he tries many things. He really does take his way out to try, although sometimes those experiments are not as spectacular as you might think, the things that he'll talk about. But he really does embrace failure and success in a very joyful manner. I think this is the one thing that, of course, the science will speak for itself, but it's extremely important as experimentalists that we learn how to fail. This one was a failure. The other thing that I do want to emphasize, which is very special about Deepak, is his joy for teaching. Over the years, I've seen him grow both not just as a researcher, but as a teacher himself, and being engaged with both younger audiences and at that same time, taking and honing on the skill of teaching. This was a picture taken in Puerto Rico with a fairly intense moment of full scope teaching. And then the last one is, things happen in the lab, as you all know. And this was an unfortunate incident with a postdoc. The postdoc did survive this incident, but he gracefully walked away from this relationship. But on the other hand, it's important to understand the playful nature that we take in outside the lab is exactly what we brought to the table doing the types of experiments Deepak will actually be describing. You will hear work that was done in the lab, out in the field, and out in the ocean. And for a single student to have the capacity to both be adept at a whiteboard and a pencil and paper, both theory and experiment, is a remarkable achievement in my sense. And one of the things that I'm very excited about is the kinds of directions that he's going to follow. And with that, I present to you Deepak Krishnamurthy on his defense. I'm going to pass this back to you.
I'm going to be laughing for the first few minutes, excuse me. But yeah, so thanks, Manu. That's really wonderful. And I'll share my screen and we will get started. Can everyone see my screen? Okay, so this is just like a little bit of a few guidelines because we are on this new medium, Zoom PhD Defense. So I think as Julie already mentioned, the question answer session will be at the end of the 1 hour talk, the public session. And during the first part, the participants will be on mute and you'll be able to unmute yourselves at the end of the talk. If you have a really burning question that can't wait to the end of the talk, we admire that. So you can send that as a question to Jasmine or maybe one of the committee members and they'll pass it on to me at the end of the thing. Okay, so with that, it's my pleasure to, okay. Yeah, it's my pleasure to be here and thanks Manu for that wonderful introduction. And as Manu mentioned, I want to echo that sentiment that, you know, this medium is not what we expected us to, you know, to find ourselves in this medium for the PhD defense, but it has its own set of advantages, I feel. And I think it speaks to the fact that I can see people from all around the world, including friends and family. And that means a lot to me. So thanks everyone for joining. And I hope you, you know, have a fun time talking about with this presentation about my science. So my talk today is going to be about multi-scale approaches to study the biophysics of parasites and plankton. And this is work done in the Prakash lab at Stanford. And this is towards a PhD degree in mechanical engineering. So to begin with, I want to set the stage a little bit to tell you the theme that underscores and underlies my PhD research. And this is basically motility across scales. So motility or controlled movement lies at the heart of life itself. And my PhD is really an exploration of how motility can impact not just at the scale of a single organism, but can have impact over much larger scales, for instance, the scale of the ocean. And I'm going to present two problems to you to kind of, you know, define and explore this theme. The first part of my talk will be on how a microscale parasite, the swimming motility of the microscale parasite, how we try to understand it, and how that motility is important in how it finds and infects humans. So that's going to be part one. And part one of this talk and actually my work in the lab directly inspired us to invent a new tool where we hope that we can make this a general measurement paradigm where we connect microscale behavior and motility of an organism to ecological scales. So that's going to be the second part of my talk where I'm going to describe this new tool that we have invented. So with that and with this beautiful quote from Rachel Carson in front of us, which really underscores the theme of this PhD, I would like to get started with the first part, which is on the swimming and host-seeking mechanics in Schistosoma mansoni cercariae. So to begin telling you about our work, I need to first set some context about a parasitic disease that we should all be caring about. So this is schistosomiasis. And it's a neglected tropical disease, which is a flatworm infection. And it currently infects more than 200 million people worldwide, mostly in sub-Saharan Africa, parts of Southeast Asia and South America. What is more, more than 700 million people live in areas where they are at risk of infection. And notably, this disease is similar to malaria in its global socioeconomic impact and disease burden. And for connecting it back to the work that I'll be talking about, the infection is actually caused by a free-swimming stage of this parasite, which is called a cercaria. And you're going to see a lot of that very interesting shape here during the first part of my talk. So can people also see my laser pointer? Okay. So this is a typical transmission site where you may get the disease based on the conditions in the water. So these are photographs from our field trip, our lab's field trip to Ghana in 2014 and Madagascar in 2016. And the reason I'm showing this picture is because you can get infected by this disease by coming into contact with fresh water, not even consuming fresh water, but just coming into contact. And this can happen during your day-to-day activities, collecting water during irrigation for cooking. And because of this, it also disproportionately affects children because of their pattern of behaviors. And this parasite actually has a fairly complex life cycle involving both the intermediate host, which is a snail, and the human host itself. So I'm going to today mainly focus on the life cycle stage between the snail and the human, and the infectious agent in that stage is actually the cercariae, which I'm talking to you about. So what happens is these cercariae mature inside the intermediate host snail, and they're released into the surrounding freshwater. And there they basically swim and find humans and infect them by penetrating through the skin. And if you were able to somehow put a microscope under a pond, this is what you would see, which you see on the right. So you would see something very interesting, which is that this parasite has this ability to stay up in the water column by active motility. So those are the red traces that you see here. So the parasite is actually swimming up against gravity. And when it's not swimming, it is actually sinking passively. And those are the blue traces here. So those are two modes of behavior that are very important. And you saw a small glimpse of the third behavioral mode, which I'll be talking about a little bit more in the next slide. So this parasite really has three behavioral modes, which you can see in finer detail here. So the tail first mode is what it uses to swim up against gravity. When it's not swimming and it's passive, it's going to sink through the water column, which you see here. And then you saw a brief glimpse of what we call the body first mode, where the parasite actually swims in the other direction. And this is going to become important later on, because this is how the parasite infects humans by penetrating through the skin. But for the purposes of this part of the talk and for my work itself, I'll be mainly focusing on what we call the tail first mode, because our measurements indicate that parasites actually spend close to 80% of their time swimming in the tail first mode, which is perhaps not surprising because this is the main motility mode they use to stay up in the water column. So without this, think of it as these are all going to sink to the bottom of the pond, river, or lake in which they're found. So they're never going to make it to the top, and therefore the chances of finding a host are probably much lesser. So we'll focus on the tail first mode, at least initially. And another very key aspect of cercarial biology that we should be aware of is that they are really, fine-tuned for finding a host, and really they don't have much of a life cycle except for that. And that's also seen from the fact that they don't have a way of getting nutrients from the water, and therefore they're energy limited. So if you kind of think of cercaria as these functionally built robots, their main job is to find and infect a human host. And they have a finite battery pack. Okay, so they don't have any means of recharging them. So once you run out of charge, you're basically going to die. And you can see that in this plot here, where if you look at the percentage of cercaria that are alive, it starts decaying very sharply around the 12 to 16 hour mark. And by 24 hours, you pretty much have complete mortality in the population. So this is going to be a very important thing to keep in mind. And this also underscores the fact that swimming motility is really their go-to thing to be able to find a host. So this is a very important aspect of this parasite, which is how is it able to find its host using its swimming motility. So given the importance of swimming motility in the disease transmission, it was very surprising for us when we first started working on this problem that there was actually a lot unknown about the first principles understanding of the swimming mechanics. So for instance, it was not widely known what is the role of this very interesting fork that you see here. So it's like this T-shaped geometry. What is the role that it's playing, and how does it actually help you propel through the water? And similarly, what is the fluid dynamical regime in which these organisms are swimming? And perhaps even more importantly, what is the swimming efficiency of this organism? Because this is directly relevant to disease transmission. So to address this knowledge gap, I'll be presenting a three-pronged approach. So the first is going to be biological experiments on the live cercaria themselves. The second, I'm going to introduce a mathematical model to understand the swimming gait. And third, we are going to have some fun comparing that mathematical model to a robotic model, okay? And then at the end, we're going to tie all of these three things together. So let's get started. So this is a high-speed video microscopy of cercaria in the tail first mode. And the reason we need to do high-speed video is because they have a beat frequency, which is around 20 Hertz. So you can't really make out the details of the swimming gait if you just image them with a normal camera. And when you're able to slow down the gait this way, you see that this fork seems to play a very important role in the swimming gait. And for the purposes of this talk, I'll be talking about cercaria in terms of three body parts, which is a very simple description of their anatomy. But I'll be talking about the body or the head, which is this part here, the tail, which you see beats back and forth, and the fork, which is actually a bifurcation of the tail. So that's what you're seeing as a fork. So we're going to be talking a lot about those three body parts. And some of you may have noticed that this gait is actually tail first. So the organism is swimming towards its tail direction. And it typically does that at a few body lengths per second. And this organism itself is very small. It's 0.5 millimeter in size. So if you look at a water column, you'll probably not even see it. It's like a speck of dust, okay? And for these kinds of swimming parameters, the Reynolds number is actually around 0.3. And one of the first things we started doing was trying to understand what is the fluid dynamical regime that this organism occupies. So what you see here is a particle image velocimetry measurement of the flow field around a freely swimming organism. And what this kind of measurement allows us to do is to conclude that based on the Reynolds number and based on these flow measurements, that the fluid dynamical regime for swimming in this organism is indeed viscous dominated, okay? So inertia, if at all, plays only a second order role, and we can think about this as a low Reynolds number swimmer. So if that is the case, it actually presents a kind of paradox, because if you notice the swimming gait, you would notice that it looks at first glance as though it's something called a reciprocal gait, where the tail is just, beating back and forth in this C-shaped gait. And if that's the case, this organism should not be able to swim. So to kind of demonstrate this very important and interesting aspect of low Reynolds number hydrodynamics, I want to show you a kind of sad video. And this video is basically a baby fish trying to take its first bite of food, okay? And you're going to see this video. So the fish opens its mouth, and then when it closes it, you see that that food particle that went in just comes back out. And you'll notice that even though the fish is opening its mouth much faster than it's closing it, the food still goes back to pretty much the same place where it started out. So this very interesting feature of low Reynolds number flows is very famously called the scallop theorem, because it tells you that if you only have a single degree of freedom, your motion is always reciprocal. And if you're reciprocal, you cannot really create a net propulsion or a net displacement at low Reynolds numbers, okay? So this is the scallop theorem, which tells you that you can't swim if you just have this single degree of freedom. So how do cercaria actually overcome this constraint and break symmetry to swim at low Reynolds numbers? To get an understanding of this, what we'll be doing is we'll be parameterizing the shape of this organism in a lower dimensional sense. So remember that cercaria have these three body parts. And so what I'm going to do is I'm going to define three bending angles that are associated to these three body parts. So we'll start with the bending angle of the tail itself that I'm shown here. And we'll then define a joint angle between the tail and the body, and another joint angle between the tail and the fork, okay? And now what I'm going to do is I'm going to look at these high-speed video data sets that we have, and I'm going to track individual parts of this organism using image analysis. So what you're going to see here is that these angles are not fixed or static, but they're actually dynamically evolving. And you can see that this organism is clearly swimming tail first, and these angles are having this very interesting pattern, which is clearly not reciprocal. So you can see that if I plot the tail body and the tail fork angles as a function of the bending angle of the tail, they actually sweep out these finite area curves in phase space. And this is your first clue that these joints that these cercariae somehow have, even though they don't have any bones, they are actually full of musculature, they actually are very crucial for locomotion. So this degree of freedom at the tail fork and the tail body joints are crucial for locomotion. So if that's the case, it begs the next question, which is that how are these controlled? How are these joints actually being specified? And right off the bat, we can think of two very broad hypotheses. One is that they are actively controlled by the neuromusculature of this organism. So the joint angles are under some kind of like neuromuscular control, and they're being specified at a certain with a certain dynamics. Or maybe life is simpler. Maybe it's a passive evolution of these joints just due to the interaction of the bending activity of the tail with the hydrodynamic drag because of the fluid that surrounds this organism. So that's what I'm going to call the passive or elastohydrodynamic hypothesis. And what we're going to do next is going to test these two hypotheses and see which one really holds water. Okay. So to begin with, we will introduce a numerical or a mathematical model to basically understand and test this hypothesis. So, this is what a cercaria looks like swimming in the tail first mode. And this directly inspired us to build what is essentially the simplest model we could think of for this T-swimmer. So we call this a T-swimmer geometry, and all it is three links which are connected together in this fashion. So the first two links are connected by an active joint, and that mimics the tail musculature in cercaria, which just bends back and forth in a reciprocal fashion. And the second joint, I'm going to first test the passive hypothesis. So I'm going to specify that this is an torsional joint, which is passive in nature. So if any of you think about low Reynolds number swimming, you would notice something very interesting, which is that this looks very similar to a very famous low Reynolds number swimmer, which is the Purcell's three-link swimmer. And remember that 3 links is important because that gives you the required 2 degrees of freedom that you need to swim at low Reynolds numbers. So with this very simple model, what you can do is you can actually write down the set of parameters that matter. So these are the driving frequency, the fluid viscosity, as well as the torsional stiffness of this passive joint. And what you will see is that we can actually non-dimensionalize this into a single parameter that we need to care about, which is the dimensionless torsional stiffness of this tail fork joint. Okay, so now we have a very pleasingly simple model. So I won't be going into the details of how we solve for this model, but the main idea is that we take the shape dynamics of this active joint and ask what is the body velocity of the swimmer. So basically, if I beat at a certain frequency, at what rate does the swimmer propel itself? And remember that the angle of the passive joint is not specified in the model. So it's something that is evolved due to elastohydrodynamics. So that's going to be the overview of how we solve the model, but we'll quickly jump into what the model tells us. And it tells us something really cool. Okay, so it tells us that a T swimmer like cercaria indeed do swim tail first. Okay, so this is a very simple approximation of what a cercaria is. And you'll notice that it started out at this baseline here, and it's actually swimming, it's propelling itself in the tail first direction. So I'm going to play that video again and point out that if you just switch that geometry from a T to an I swimmer, you have essentially a reversal in swimming direction. Okay, so you started going tail first as a T swimmer, but you start going body first as this I swimmer that I just showed you. And what is even more remarkable is that nature already beat us to it. So I remember showed you the 2 behavioral modes in cercaria where they're active. So in the tail first mode, of course, they swim tail first, as you've been seeing so far. But what I didn't show you is that they can actually transition extremely rapidly from this tail first mode to a body first mode by doing exactly what our model just showed, which is to just close their fork and transition to an eye swimmer. So what this means is that this parasite already kind of exploits this very simple morphological transition that gives you a very different behavioral mode. And indeed, this behavioral mode is very important because this is what cercaria use to penetrate through the skin when they are already very close to the body. Okay, so this is very important. And remember that this is a slowed down video. So cercaria can switch between this in a few 100 milliseconds or lesser. So this model kind of, piqued our interest to an extent that we really wanted to go ahead and, being experimentalists, like see it for ourselves in an experiment. So what we did was build a scaled up model of this T-swimmer as a T-swimmer robo. So this is what you see here. And this was one of the first like experimental projects that I did in my PhD. And this was a lot of fun because, I had to make this robo completely free. So you couldn't have wires going to it because that would affect the measurements of the swimming velocity. So we made this system that has an onboard actuator and a battery and sensing so that you can specify a very precise joint angle. And this was actually work done with a very talented master student, Arjun, who was in the lab at that time. And so remember that this T-swimmer robo is much larger. So that's my hand there. So it's around 10 centimeters in size. But I told you that the cercaria is only 0.5 millimeter in size. So how do you actually compare the fluid dynamics of swimming of these two things? So it turns out you can do that by just changing the viscosity of the surrounding fluid. So another fun thing I got to do in the first part of my PhD was to fill a huge tank of, aquarium tank with around 30 gallons of corn syrup. So this was a lot of fun to put in. It was not fun to clean out. I can. So this was our corn syrup swimming pool. And what we used to do is put these robos in them and basically see how they swim, okay? And so what I'm going to show you next is basically a race between 5 different robos. So remember I told you that the main parameter we need to worry about is the joint stiffness of this tail fork joint for this 3-link swimmer model. And so here what I'm doing is I'm going to change the joint stiffness all the way from 0, which is what I call a free joint, all the way to infinity, which is like a fixed joint, okay? And what we are going to do is increase it logarithmically in between those two values. And we're going to raise these robos against each other. And if you watch this for a bit, you'll see something very interesting. So you'll see that the free jointed robo and the fixed jointed robo are terrible swimmers. They never go anywhere. They're stuck at the start line. But all of these intermediate stiffness robos actually are good at propelling themselves. And what is more, they propel themselves in the direction that the model predicts, okay, which is nice. And so both model and experiment also show us that there is an optimal stiffness of the fork joint based on the frequency that you beat your tail at, okay? And that's what you see here. So if you plot the average swimming speed of the organism as a function of this tail fork joint stiffness, you see that there's a single well-defined peak where you maximize the swimming speed. And you can see that not only in the mathematical model, but also in our scaled up experiments. So now we have an understanding of the fact that, this 3-link swimmer already kind of gives you the qualitative aspects of cercarial motility. But now you're going to do one step further, which is we're going to add back the 4th link that we had initially ignored. Because remember that we didn't talk about the body tail joint in cercaria. So now we are going to add a 4th link. And so we're going to have what is essentially a 4-link T-swimmer. And because we have a 4th link, we need to add one more torsional spring here, okay? And we also found a way to measure these torsional stiffnesses in the actual organism using biophysical experiments. And with that measured value, we could actually directly compare the mathematical model with the biological experiment. And you can see by looking at this video that you already see a good degree of qualitative similarity between the gait of this T-swimmer model and the actual experiment. And what we found is this similarity goes beyond just a qualitative sense. You can actually quantitatively compare the displacement of the model and the displacement of actual cercaria. And you see that the model does pretty well in predicting this displacement. And not only in the displacement of the entire body of the swimmer, but even if you look at the shape dynamics. So for example, if you look at the tail fork and tail body angles as a function of the bending angle of the tail, you indeed see that the model is very good at kind of predicting both the quantitative extents and the qualitative features of this shape dynamics. So this gives us a lot of confidence to say that, pending any other data that tells us otherwise, we can safely conclude that we don't really need an active neuromuscular control to explain the current data that we see in cercarial motility. And it's very likely that it's actually a completely passive elastohydrodynamic system that these organisms are using to propel themselves, which is quite elegant. So because we have access to this insight and this mathematical model, we can also ask this very interesting question of how far away are cercaria from what is the theoretically most optimal swimmer you can make at using this geometry. So here what I'm showing you is the average swimming speed measured as a function of tail body and tail fork stiffnesses for the mathematical model. And you can see that the model predicts this single peak here in this phase space. And very interestingly, cercaria are not at all very far off from that peak. And you can see the same trend for swimming efficiency. And what this really goes to show is that perhaps unsurprisingly, given the kind of evolutionary pressures that are on these parasites to transmit to their host, it's not surprising that they actually have this kind of extremely well-tuned swimming motility to swim efficiently and fast. Okay, so with that, I want to give you a quick summary of the first part of my talk. So I've told you about this very interesting swimming gait in cercaria, which is using this fork tail geometry to as a reverse gear, basically. So cercaria are using it as a reverse gear to swim up against gravity. And because they use it to swim up against gravity, this has definitely implications to the ecological distribution of these organisms, because it allows them to stay in the upper layers of the water column. So I've also told you that this gait is simple and robust in the sense that it does not involve any active feedback or control. So it's just a single driven oscillator that is giving rise to this behavior. And because we have access to this model, we also get to do some interesting comparisons between physiological parameters such as muscle stiffness and swimming motility parameters such as swimming efficiency, which is for the first time. And this becomes even more interesting in the light of recent papers where people have looked at new compounds that actually paralyze cercaria and prevent their infection in mice. And the way that that happens is still very interesting and an open question. And it'll be fantastic to compare our models and insights to the action of that drug, and also use it as an insight for novel drug targets. So, this was work that was done in the lab. At the end of my second year of PhD, I got this fantastic opportunity from Manu, who told me like a month before my wedding that I could go to Madagascar if I so wished. And so I had a very nice decision that I had to make. It was not stressful at all, but I made the decision to go to Madagascar, and it was probably one of the best decisions in my life. Thanks, Manu, for that opportunity. So we actually got a chance. Oh, before that, I should not forget to thank my co-authors for the first part of the study. So Yorgos, who was a PhD student, and MI instrumental, both mentoring me in the first part of my PhD and also teaching me how to do this really amazing image analysis you saw to kind of extract the bending angles of each part of the cercaria and so on. I kind of showed that very quickly, but there's a lot of work that just went into how you do that. And I already mentioned Arjun, who helped me with the experimental parts of the T-Swimmer robo, as well as Professor Judy Secanari from UCSF, who provided lab space and organisms and really got us started. So with that, I want to talk a little bit about this experience in Madagascar. And our goal here was actually, at least my goal was very simple, which was to really see the disease in its ecological setting, both in terms of its ecological setting in case of in this case, you can see that we are collecting snails near an irrigation canal. So you can see that's Manu in the water there. And this is Andres, who was our local collaborator. And we also worked with a local medical student, Aina. And our goal here was to collect snails that may carry Schistosoma cercariae and basically bring them back to the lab and study them at a scale higher than we had so far. So remember, so far my view has been very microscopic. So now we want to zoom out and kind of ask how that microscale motility interacts with, for example, the distribution in the water column. So this is kind of what our effort was really geared towards. And interestingly, at the end of 1 week, we actually found 3 different species of cercariae, not of human parasites, perhaps thankfully, but of parasites that infect birds. And so this is an example of a cercaria that is being shed from a snail. And indeed, actually, the snail species is common to what would also carry the human parasite. And our goal really was to image these organisms in a vertical column of water to mimic the kind of environment they would see in the natural setting. And we built this kind of interesting rig to image the density of these organisms along a vertical column. And you can see that in action here, and you can see the kind of data sets that you get from it. So here I'm actually injecting a bolus of cercaria into this instrument, and you can start seeing immediately that the swimming motility has very clear consequences to their vertical distribution. And you can see that over time that this vertical distribution both spreads out and also sinks lower, which is kind of fascinating because we now have insights at both microscale and this macro scale. And while preparing for this trip, Manu and I were kind of sitting in his office and perhaps doing the things that we were not supposed to do, which was to actually plan for the trip. So we were actually brainstorming ideas to actually study this kind of behavior over, say, a meter, but without losing the resolution at the microscale. So I want to motivate that by kind of saying that we really wanted to link behavior to ecological dispersal. And we wanted a behavioral assay that doesn't sacrifice scale. So remember, when I just showed you that data set, I can't really make out what individual organism is doing at the microscale. And so we tried to kind of imagine a system that could measure, for example, cercarial behavior over its entire lifespan of 24 hours, but without sacrificing the resolution at the microscale. And this actually gave rise to an idea which kind of became the second part of my PhD. And it will also be the second part of my talk today. And this is what we call scale-free vertical tracking microscopy, or gravity machine for short, okay? And this is our efforts to do exactly what I just said, which is to connect microscale insights, say cell biological insights, to macro scale phenomena, maybe at the scale of the ocean, okay? So remember that this is a very tall order, literally, because you're comparing 100 micron size organisms, and you're asking what they're doing over 10 meters, which involves like at least 5 to 6 or maybe even 7 orders of magnitude. And while we were motivated initially by our work on parasitology, we very soon recognized that this is actually a very, very important problem in the ocean, okay? So in the ocean, life in the ocean is, we see, for example, big mammals like whales and things like that, but there's a whole world of the ocean that is hidden to us, and this is in the form of microscale plankton, okay? And even though these are very small in their size, they have a very large impact on planetary scale biogeochemical processes. And one very simple example of this is that marine primary producers, so you can think of them as the plants in the ocean, though they are actually nothing like plants, they actually fix 50% of global carbon. And they give rise to, for example, these fantastic blooms here that you can see from outer space. So I'm talking about a single cell that is blooming to the extent that you can actually detect it from outer space. And interestingly, we don't really think of these when we talk about, say, carbon fixation, because we typically are biased to think about terrestrial rainforests and terrestrial ecosystems. So I want to tell you that these microscale plankton actually have a very, very large impact both over our own lifespans, but also over the lifespan of the planet. And a remarkable example of this is the chalk cliffs in Dover, which you can see, which are actually made up of the shells of these phytoplankton, which is just kind of mind-blowing to just think about. So Another very important feature of marine ecosystems that makes the kind of tool that I'm going to talk about very relevant is that marine ecosystems are vertically structured. And this is simply due to the effect of gravity and light scattering, that the ocean is a stably stratified environment for the most part, where hotter and less dense water sits on top of cooler, denser water. And most of the parameters that life cares about change much more rapidly along the vertical direction than they do along the horizontal plane. And perhaps unsurprisingly, life in the ocean reflects this underlying nature of their ecosystem, which is that every single day, microscale plankton, all the way from single cells to small organisms and all the way to fishes, have this amazing ecological phenomena called diel vertical migration, where they actually migrate vertically through the water column over length scales of 10 to hundreds of meters. So this is, in many cases, 1,000 to 10,000 times their own body size every single day. And this, as well as the sinking of dead and decaying matter in the ecosystem, which makes up a very large and very important aspect of the vertical material fluxes in the ocean, this kind of gives us a picture that microscale processes at the scale of single cells, small organisms, and sinking particles, evolving in a vertically structured environment, really shape marine ecosystems. Okay, so I want you to kind of keep that in mind for the rest of the talk. And because we are interested in microscale processes, which are happening along the vertical axis, we are going to shift our perspective. So I know most of you who have looked at a microscope or have used a microscope probably used it in this kind of configuration. So you put sample on a cover slip and then you look through an eyepiece or a camera, but the sample is in the horizontal plane. But for the purposes of this talk, we are going to shift our perspective to the vertical axis. Okay, so we are going to now image along the vertical plane where gravity plays a role. So this is not a new idea by any means. Vertical microscopy is something that people have cared about for a while. I'm going to quickly summarize the state of the art and kind of show you where the gaps lie. Okay, so the goal in vertical microscopy is fairly simple, which is that you want to follow a microscale object and see its behavior along the vertical direction without sacrificing the microscopic resolution. Okay, so the simplest thing you can do is perhaps this, which is you have a fixed camera. So you have let's say a tall column of fluid, and you have a fixed camera looking at this fluid from the side, and say you have an object that's going past your field of view. But the problem, remember, is we are doing microscopy, which means that the fields of view are typically very small. And this means that the track length that you get of something going past your field of view is typically a few millimeters. Okay, and this is not good because it really biases your data to things that don't move much at all. So things that are going to spend a lot of time in your field of view are going to be what you're going to collect most of. So you can do one better than that if you want, which is to move your camera or your optics or move your stage. Okay, and there you can collect longer tracks, definitely, but eventually you're going to hit the wall. Okay, literally. So what that means is that you're still very far away from the kind of scales that we were just talking about, which is hundreds of meters. And that's, you should really be thinking of, if you're from San, find your nearest skyscraper and look at it. And that's the kind of scale that you're really talking about in the ocean where these processes are evolving. So you're at least 3 orders of magnitude away from accessing those scales. And that kind of gives us a very simple problem statement, which is that how do you image a freely moving organism at microscale resolution, but traveling ecological scales in the vertical direction? And of course, you could do something. If you had a lot of money, you could buy a skyscraper and like put a really tall column of fluid and buy a really expensive microscope and build this crazy contraption that can go back and forth and track a single cell. But we didn't have that, thankfully. So we had to come up with something slightly simpler. So we came up with a very simple idea, which is that you can take a tube and bend it back on itself And what you get is this, you get a circular fluidic chamber. So you get like this annulus of fluid. And this is a poor man's water column. It's an endless water column if you don't have access to a skyscraper, okay? And this idea is very interesting because now you can do microscopy at these two locations, which are special because gravity is along the tangent of the circle there. And if you image at these two locations, you get what is essentially a microhydrodynamic treadmill for single cells. And the idea is again very simple, which is that now when you have a small object, say a single cell, moving vertically in that location, you can compensate for that motion using a closed loop tracking system by rotating that fluidic stage in the opposite direction, okay? And then what you get is that the cell actually never goes anywhere in the lab reference frame, but the cell is actually moving through the fluid as though it's swimming in a vertical column of water. So you can map the angular displacement of this rotational stage to the virtual depth that you're measuring when you track the cell. So to take that from an idea to a realization, we had to do a lot of things. And we also decided that we were not satisfied with just tracking along the vertical direction, because cells can also have interesting movements on the horizontal axis. So what we built is essentially a 3D tracking microscope where the vertical axis is tracked using the circular fluidic microhydrodynamic treadmill, but the horizontal tracking is a regular linear stage. So you can track in both X, Y, as well as in Z. So Z lies along gravity. And remember that we are doing this in a closed loop fashion. And what the simplest way I can explain this is that imagine you are on a treadmill that could somehow automatically adjust to your walking or running speed. So let's say you are walking for some time and then you instantaneously started running very quickly. The treadmill would be able to adjust and still keep you on the treadmill and not make you fly away and fall and like hurt yourself. So that's kind of what this microscope is doing. It's based on the behavior of the microorganism, it's automatically sending motion commands to the stage to keep it always centered in the field of view. So this is what the microscope looks like in its close to current form. So if you come to the lab, this is kind of what you would see. And you can see the circular fluidic stage here. And you can kind of I'm talking about the light microscope, which we have built here. And currently this is built for both dark field and fluorescence imaging. And I'll talk about that a bit later. And going from this idea to a realization was actually a really fun and I think a very defining movement of the second part of my PhD, because it was actually a technical challenge on multiple levels. And I'm going to talk about just one of them today. So just for the lack of time, and that one is actually one of the interesting ones because it comes really from this unique geometry that we have, where we have a continuous annulus of fluid with no boundaries in the direction, because that's really key towards our idea, which is that we don't really have any walls that the organism can bump into. And what we did is we realized that in that geometry, if you move the stage and you change the velocity of the stage, there is actually a certain amount of fluid dynamics that happens that we need to care about. For example, the simplest description of that is that when you move the stage, the fluid actually suffers a delay in coming to the final velocity of the stage. And we need to actually worry about that when we track, because we want tracks that are as high fidelity as possible to an actual swimming organism. So to actually understand this better, we actually developed a mathematical model, and we derived what we call a tracking delay condition. And it really involves 4 time scales. So one is the time scale of the object that you're tracking, which is how fast the object can change its velocity. The second really is a function of the microscope that you're using. So if you use a 100x objective, you typically have a much smaller field of view. So the organism would swim out of your field of view much faster. And the last 2 time scales are related to the microscope stage and the fluid itself. And this is how long it takes to kind of move the stage and move the fluid. So you can think of these as time scales that give you that delay. And if you actually write down this mathematical model, you get a very nice and kind of pleasingly simple relationship, which is what you see here. And what this lets us do is to make design diagrams for actually speccing out this microscope. So for instance, so let's say you wanted to track an object that was moving at a certain speed and it's capable of changing its speed at a certain rate. You could ask yourself, how big a chamber do I need to actually track this object reliably? And so what you would do is you would generate this plot and kind of locate yourself on this plot. And if you are on a positive side of this plot, the probability is very high that you'll be good at tracking. And if you're on the negative side of this plot, there's a high probability that you may lose the organism very frequently. So that, you know, gives us a very nice way of like thinking about the design space of this instrument. And we have done this for several other parameters, which I won't be going into today. But the basic takeaway I want you to have is that there is actually a very large region of parameter space that we can use to track microscale organisms, all the way from a few microns to a few millimeters. And in some sense, that need not have been the case, right? So I mean, it could have been that this whole idea was very cool, it's very nice, but it really doesn't work in the real world. But, you know, thankfully it did. So, you know, that's something that we can actually show mathematically using these diagrams. And one of the first, you know, most simple experiments you can do with this instrument is, you know, track a sinking microsphere. So here I'm going to show you that if I track, you know, a sinking sphere using our method, you know, where the sphere doesn't really go anywhere, it's actually in the same location, and it's the treadmill that is rotating. And in the other case where, you know, you're in an Eulerian framework where the bead is actually going past your fixed camera, you would expect, as, you know, based on what I've told you, that you would get the same sedimenting speed in both the cases. And that's exactly what you see. And this is for over different bead types and bead densities. So now that we had this, you know, basic calibration and like we have a working microscope, actually the first thing that we did as soon as we, literally this is not even a joke, the first thing that we did as soon as we got this microscope working was to take it apart, because we had to take it to Hopkins. And, you know, we got this fantastic opportunity in 2017 and then also in 2019. And this is something that we have done several times, which is that we have taken this tool to the location where the most interesting samples are, because they are not always in the lab. And especially when you talk about samples from the ocean, there are many things in the ocean that don't really like living in the lab. And so you need to really take the lab to the ocean for this case. So you see here, you know, us kind of building the first version of this instrument in Hopkins back in 2017. And, you know, this was really crucial. You'll see a lot of the data that I'm going to show you is really from measurements that were done at marine stations and at sea, okay? And I mean, it's always a cause for excitement. You can see people crowd around the instrument and it's always fun. So one of the first things that we did was, when we had this tool working, was we tried to build the first database of multi-scale plankton behaviors. So remember, I told you that we now have a tool that does not sacrifice scale. So you can get both this kind of microscopic resolution. So you can see that these are marine invertebrate larvae on the left and single cells on the right. So this itself has like around 1 order of magnitude in length scale. And we are also tracking these organisms in 3D space. So you get these trajectories that you see here. And what is remarkable, and what kind of already motivates why this tool is interesting, is that, for example, when I'm tracking this polychaete larva here, which is going to come back later in the talk, this larva actually is capable of swimming 0.3 meters in 5 minutes. So just think about that. This larva is around 250 to 300 microns in size, but its motility is so directed and so rapid that it actually covers these pretty tall regions of space in a surprisingly short amount of time. And you actually see that even for single cells, okay? And all of these tracks are over the same amount of duration. So it kind of also shows you the kind of behavioral diversity you would see in these organisms. So in the next few slides, I'm going to be switching between many different organisms. So I'll just say this, it might be a bit overwhelming, but don't worry about that. It is by design that we actually imaged a huge number of organisms to start with. And so we're not going to be focused on one organism like I did in the first part of the talk. So kind of brace yourself for a few different organisms to come your way. We will start with one of our favorite organisms in the lab, which is the larvae of the bat star, which is Spateria miniata. And this is a starfish that you can see very easily off the coast of California if you go to tide pools. And here we are looking at the behavior of the larval form of this invertebrate organism. And this larval stage is it's only 750 microns in size. So it's much, much smaller than the adult. And what is one thing to remember is that these invertebrates, before they become, the kinds of organisms that we see in the tide pools, they spend a long time in the water column in this plankton, okay, in this planktonic stage. So it's very relevant to be asking what behavior, what does a Pateria miniata larva do in the ocean? Like I think you can safely say that we don't know. So this is kind of, trying to address that gap a little bit. And this data set is fun because it also shows you few aspects of the tool. So here you start by looking at the larva. It's swimming straight up in a very directed sense. And now I'm going to slow down the movie a bit to kind of show you another capability of the tool, which is that because we have this micro scale resolution, we can actually measure the flow field around these organisms during behavioral transitions. So you now saw that the larva went from swimming straight up to sinking, and it creates this beautiful region of vortices around its body. And this is actually a feeding behavior. And you see that it continues to sink in this very graceful fashion. And eventually, at the end of this video, you'll see that the larva actually goes back to its upward swimming trajectory. And even in this video, you see something very interesting. That's not the only time that behavior occurred. It has been happening at a certain frequency. And one very interesting thing is like how repeatable is that behavior? Because we have scale-free tracking, we can literally ask this question and answer it directly. So for example, I'm showing you a data set of the Z displacement of a single organism now measured over more than an hour, okay? And there are a couple of things that are very interesting about this data. Firstly, just kind of look at the vertical scale. That's 2,500 millimeters, which is 2.5 meters. So if I was standing up and giving this talk, it would be taller than me, okay? And not only that, you see that behavioral transition happening repeatedly over the scale of this tract, which is quite remarkable. And because we can measure this behavior so repeatedly, we can actually ask, is there a universal profile to the swimming behavior of this organism? And it indeed seems like that's the case. So if you now average that behavior over 25 such transitions, you see that the larva always switches from a swimming up mode to a period of sinking, and then transitions to a period of hovering. And so that prototypical behavior is quite interesting because it now begs the question, how is that being controlled? What is the clock that this larva is using to kind of, know that it has to feed at this certain interval? How does the environment affect it and so on? Okay, so we're going to switch organisms now and go to the polychaete larva, which I showed briefly before. And this data set also gives us the first glimpse of diel vertical migration, but happening at the scale of a single planktonic organism. So this is a polychaete larva swimming down, and this is during a daytime track. And if the same organism is imaged at night, you see that they actually transition to being upward swimming, which is exactly what you would expect from a diel vertical migration sense. And you can see that if I now measure this over several organisms and over several tracks during the day, you see that the daytime tracks always kind of fall in this negative velocity bin, and the nighttime tracks always fall in this positive velocity bin, which is quite remarkable. Okay, so we'll switch organisms again. And I want to tell you about a very unique aspect of this tool, which it lets you do virtual reality experiments with single cells and plankton. So we're going to put VR goggles on plankton now. So far what we have done is basically imaged plankton in a zero information environment. So this is kind of like if I gave you VR goggles and I didn't show you anything, and I kind of showed you like what would look like deep space, right? You're floating around, you have no reference point pretty much except gravity, right? And now we're going to add in, cues that these plankton may see in their natural environment. And this is actually a very powerful tool that people have used in behavioral neuroscience, where you can actually have observation of a fly brain and look at the signaling of this, and then at the same time, see what the behavior of the fly is by tracking it on this, what is called a fly ball tracker. So you can kind of see the similarity to our approach here. And now what we're going to do is bring a technique which you can do this for vertical gradients. So now imagine that you have a spatial gradient, say in light intensity, as a function of depth, we can now map that to a temporal gradient in this fashion, where based on the behavior of the organism, we can slowly increase the light level so that the organism actually thinks that it's coming closer and closer to the surface of the water. So this is a very unique kind of experiment that we can do with this instrument. And so basically it's a mapping between spatial and temporal gradients, which are driven by the organism's own behavior. And to give you a quick example of this, I'm going to show you a light gradient, which is very artificial. So not like the natural one that I've showed here, but where the light actually changes, it flips on or off every 20 millimeters. So it's kind of like I was kind of playing with the organism, and every time the organism swims up by 20 millimeters, I'm going to flip the light intensity. And that's what you're going to see here. And this is in an organism called Volvox, which is an algal colony. And over the scale of this track, you don't really see much, right? It just feels like this colony is swimming up. But the reason you don't see much is because that scale is actually a meter, okay? So this organism is 500 microns in size, but this vertical scale is a meter. So what we now can do is actually zoom in and ask what was actually happening in each of these transitions. And what we find is very interesting. So if you go from a dark to a light transition, we find that this organism actually pauses for a bit, almost like it's not sure what to do, and then it keeps going up. But if you look at the same behavior from a light to dark transition, as you're going to see here, you see that the organism doesn't seem to care. It just keeps swimming straight ahead, okay? So this is the kind of, kind of environmental tweaking that we can do, and we can really talk to the organism and kind of ask it, like, what is it that you care about? So I'm going to end at least this part of the talk with one very interesting biological puzzle that we discovered on a field trip to Puerto Rico. And this is when we were tracking single-celled diatoms. So remember I told you about marine primary producers. Diatoms are probably one of the most important classes of marine primary producers because they have a pretty large impact on global carbon fixation. And the sinking of these diatoms are very important because they actually directly can set the vertical fluxes that is involved in an ocean ecosystem. And so what you're going to do is image this organism. So I'll tell you one thing about the organism is that it has really no means of swimming. So it doesn't have a cilia or a flagella. And so when we started imaging this organism, we were actually in for a shock. So what we saw is that this organism indeed does have some form of motility. So you can see here a single cell choosing to be either a sinker or a floater, okay? And it's making this decision over extremely rapid timescales of a few hundred milliseconds. So you're going to see the cell basically come to a complete halt. So it's now almost the same density as the surrounding fluid. And if you keep watching this data, you're going to see that at some point, it's going to very sharply transition to a period of extremely fast sinking right there, okay? So you started sinking right there. And because we can measure this over long times, we can actually derive this kind of universal trend that we see is that there are literally two states that the cell occupies. And so it's probably, it's actually transitioning between these scales, between these states alternatively over the course of this track. Okay, with that, I want to summarize the second part of my talk, where I've kind of introduced a new tool where you can do multi-scale measurements spanning microns, milliseconds, all the way to meters and days. And this really opens up a new paradigm for measurements, both in the ocean as well as in other systems. And this has already shown us that plankton may not just be drifters, and behavior is very important, especially along the vertical direction. So as our polychaete larva showed us, even over modest timescales, the kinds of displacements that we measure are quite remarkable. And I also showed you this virtual reality paradigm for single cells, which are freely swimming, where we have this very interesting coupling between cell physiology at the micro scale, cell scale, and environment over ecological scales. And as I was mentioning before, a lot of this is also done. Recently, we got this chance to go to the ocean itself, not just to a marine station. And we took the instrument on board this research vessel Kilo Moana in last December. And this is just a small example of how we can actually collect this kind of data, not just in the lab, but on board a moving research vessel in the North Pacific. So this is just a simple example of what it looks like. And to give you proof that that's in the ocean, that's what it looks like outside the window. Okay, so with that, I also want to have a little bit of look towards the future and tell you that this tool really opens up measurements for problems, not just in the kinds of things that I've talked about, but also in fundamental fluid mechanics, marine snow dynamics and microplastic sinking rates, plankton dispersal, which I've mentioned a little bit, as well as bringing it back to the, what started it all. So basically coming back a full circle, essentially, which is to really measure parasite behaviors and biophysics over longer times and start asking, can we really connect these behaviors to ecological phenomena? And one of the things that we are very excited about is that we want to do this in a fashion where anyone who has a wide field microscope can literally put their microscope on the side and drop in a stage, which is basically a gravity machine, and then have this kind of vertical scale-free tracking capability on a regular microscope. So we are calling that Gravity Machine X to kind of bring this to not just custom microscopy, but also to traditional microscopy. So if you're interested in finding out more, definitely take a look at our website, www.gravitymachine.org, where we have a lot of these data sets that I showed you, so you can explore those data sets and also look more into the technical details of the instrument, as well as the kinds of things that led to it being conceived in the first place. So with that, I'll thank my co-authors for the second part of the talk. I mean, I'm really lucky every day to call these people my colleagues and friends. Hong Xuan, Adam, Ethan, as well from Prakash Lab, as well as interns from France who were part of the Prakash Lab in previous years, Delphine, Francois, as well as Pierre, as well as an extremely talented resident artist, Rebecca, who's responsible for these jaw-dropping graphics that you saw in the first part of my talk. And that brings us to the end of the scientific, maybe the technical scientific part of the talk. And if all of you will indulge me for a few more minutes, I will quickly thank the people who have really made it possible. And I think this is as important as talking about the science itself for me. So I'll quickly, first and foremost, thank my amazing committee. So Professor Sanjeeva Lele, I want to start by thanking you. I've known you from literally my first quarter in Stanford when I took your course on instabilities and waves and fluids, which was really fun and kind of a defining moment of that first quarter. And I've since taken many other courses with you, and it's been just amazing to have your mentorship and guidance, both in the fluids aspects of the project, but also in general. So I want to thank you for that. And another person who I've had the pleasure of knowing for a long time is Giulio. And it really gives me a lot of pleasure to say that, like, I met Giulio in my second quarter in Stanford when Manu told me suddenly that I had to attend this meeting, which is I think a Gates Foundation Schistosoma meeting. And actually that was one of the greatest experiences in my whole PhD, I'll say. I learned so much in a single day and met so many people that I couldn't have imagined. And it really, started me thinking about not just the microscale biophysics, but also the ecological scale problems in schistosomiasis. So Giulio has been just a fantastic mentor and a supporter and a tremendous resource in terms of thinking about the ecology of this parasitic disease, and the kinds of approaches that he's taking are just super inspiring. So I want to then thank Professor Bo Wang. So I'm again very lucky because Bo's lab is really right next to Manu's lab in Shriram Center. So I get to interact a lot with Bo's lab members as well as Bo himself. And it's been fantastic. Bo's been a fantastic resource in talking about schisto in general and in general in molecular biology. So thank you so much, Bo, for your help and support. And I also want to thank Professor Mark Denny, who first, when I first visited Hopkins, I really felt had literally the world's best office view. So this is kind of what you would see from Mark's office. I don't know if that's where he is right now, but that's like, this is amazing because Mark's really been an inspiration in thinking about biomechanics from an ecological perspective. His work in that field has been really inspiring for me. And recently, he's been a great source of support and as well as feedback for our work as it starts becoming more and more relevant to the ocean. So thank you so much, Mark, for your support. So that brings me to Manu. So, I mean, I'm not going to really try to say everything I want to say about Manu, but thanks, Manu, for I think what's really been like an opportunity of a lifetime. I think Manu is really inspiring for the fact that he really made me come to terms and be at peace with the fact that science is a human endeavor. It's not just, it involves both the heart as well as the mind and the brain. So thank you for kind of leading by example, showing me that every day and through the lab itself. And my relationship with Manu has always been this, I always question the crazy ideas or counter it with an idea of my own. And someone at the end of the day needs to stand on top of the desk to kind of prove the point and prove to someone that what is actually really happening. So this is basically Manu standing to show us that I think soap films don't break under certain conditions, which I don't know. I think it's still an open question. So, and I think it's been really a joy of discovery and kind of, bringing our hearts to science every single day. And thank you for showing that by example. So I can't say how valuable it is to kind of find a home for that kind of thinking. So thanks, Manu. And that brings me to the Prakash Lab. So again, I'm not really going to try to say everything that I want to say, but I really can't ask for a more fantastic, more generous, and more brilliant set of people who are somehow in that basement in Sriram. That's just, to me, mind-boggling, that every single day I get to come and talk to these people and joke around with them and do some science. And that's really been, the defining aspect of my PhD has been not just interacting with my PI, but the lab itself. And I'll say that, yeah, thanks for everything. And I think we've had too many adventures to put on this one slide. So I've tried my best, but the pot is always boiling. So with that, I also want to thank a very special aspect of my PhD, which has been that I've had this opportunity to both mentor and learn from a fantastic set of interns, both from France and in Stanford. I'm just highlighting a few of them here: Delphine, who was here last year, and Francois, who was here in 2018, and Pierre in 2017, and Fabian in the year before that. So I really look forward to my summers in Stanford during my PhD, because it means that I get to work with another fantastic, kind, generous, and brilliant person who somehow lands up in the lab. And I've, to be honest, I don't know what they learned from me, but I learned a lot from them. So thank you for being here. Some of them are actually here, so thank you for that. Really value that. That brings me to the larger Stanford community and the people who have really made this PhD possible and have kind of oiled the wheels and kept it running smoothly. I want to really thank Prakash Lab admins, both past and present, Christine Kurihara, Jasmine, who's on the call. I also want to thank, from the bottom of my heart, the admin staff, both from my home department in Meki as well as my adopted department in BioE, for all their help at several crucial junctures. I also want to thank Vinny, who's our building manager at Sriram, for really making it all possible, and for making sure the bricks stay in place, and for his spirit and fun personality also. And there's a story behind this, which I won't get into. And I also want to thank two very special people from Hopkins Marine Station, Ayelet and Kathy, who were just fantastic hosts for us last summer, and who made last summer both nationally for both me and many people from Manu's lab. So thank you. And I also want to thank Professor Akribos, who I think is on the call, for just being an incredible mentor and just a source of inspiration by just talking to me. So thank you for that. And there are definitely people I've missed out here, but thanks to everyone who made it possible. that brings me to where it all started. So I want to thank my parents for everything. I mean, I think, again, I'm not going to say everything that I want to say here, but the books that you see are actually very special because these were actually the first books that, really made me think about science a little bit differently from, say, what the textbooks were teaching me in school. And these books were really a big part of my life because they told me how fun it is to build with your hands. And that's been a source of joy for me for my life so far, and I think will be for the rest of my life. So, thank you for making that possible. They are on the call. So it's fantastic that, you kind of, I don't know, piqued that interest so early on. So I want to thank you a lot for that. I also want to thank my brother, who's also on the call, for being my partner in crime and kind of being like the person who keeps me intellectually honest in general. So thank you for that. So that's the both of us performing together at my wedding. I also want to thank, so I'm very lucky to have a second family, basically. So I got married through in between my PhD and my in-laws who visited me a few years back. This is a picture from then. So they're also on the call. So thank you for taking an interest in my science. I don't take that for granted at all. So they, for example, know what a trichoplax is. So Manu, they're going to ask you some questions. I also want to thank my grandparents. I think two of them are on the call. So this is my Patupati here on the left. She's one of the first audiences to my poster, which is on the biophysics of parasites, which is actually fun. And I also want to kind of highlight this very special instrument, which I use every day in the lab, which incidentally comes from my grandfather, which is, he was not a scientist himself, but he somehow saw fit to buy this beautiful vernier caliper, and I get to use it every day, which is kind of a special meaning for me. I also want to thank my other grandmother, who's not here today, but thanks for just being a supporter. And finally, I want to thank my partner Preeti, who's on the call. I think just for being a pillar of support and just for being someone with whom everything is an adventure. She had to leave the country in the middle of my PhD. But we, kind of made it into an adventure. Since I do some theory, I used to just go there and just sit and do pen and paper work. And, we used to go on adventures together. And literally that's what happened. And yeah, just thanks for everything. So again, I'm not going to say everything I want to say here, but yeah, I value the support that, I know doing science sometimes can take a toll. So thanks for all the support. And with that, I want to thank the audience for your attention for over this long time. And I can take questions. Yeah.
Well, fantastic. Yeah. So a virtual applause. Great presentation. Really moving acknowledgement. Thank you very much, Deepak. I think we have about 5 minutes for a little bit longer for questions, depending how many of them. So I open up. I'll try to monitor also the chat in case there is anything. And
yeah, people who have questions, if you could write questions in the chat. And I think the first question is from Janet Yen. Jasmine, can you unmute Janet?
I think I
Georgia Tech.
I think everyone should be able to unmute themselves right now.
Okay.
I think I unmuted myself, and I think, can you hear me?
Yes, go ahead.
Okay, but I think I also connected the video. Okay. Anyway, Deepak, that was a terrific talk. It's so much fun to join you from Atlanta. I mean, I actually think this COVID thing might have changed the way we speak to our colleagues. It's just so much fun. But anyway, of course, I'm going to ask the question you expect. So what is the largest plankton that can be visualized to study their swimming behavior and the way they modify the flow around them? Like, can you do copepods or jellyfish?
Yeah, thanks, Janet. Yeah, that's a great question. So I think I was trying to allude that a little bit in that design diagram, which is what you see here. But we have by no means pushed the limits of, I think, what this instrument is capable of, especially at the larger scales. And I think for example, the copepods that you sent us kind of already pushed us in that direction, which is that we have built this so far for more tuned towards microscale plankton, but essentially this would involve scaling up the entire chamber and also having like lower mag optics, which will be still sufficient to image things of the scale of copepods and jellyfish. So of course, the other thing that matters is the kind of behavior. So copepods are challenging inherently because they do these jumps. But we also have this other method of tracking both at low and high mag, which I think can be like fruitfully applied to these more jerky kinds of motions, both in copepods and maybe other plankton. So I think it's basically, think of it as like a design that we would make, which is tuned for larger scale plankton.
Thank you.
Yeah.
Other questions? And then if you could write your name in the chat, that way we know who might be asking questions. There's a question from Paul. Paul, do you want to go ahead?
Sure. I'll sort of repeat the question, which is that I was curious how you lit the gravity scope to consider sort of the deal migration, and also, yeah, were you able to incorporate sort of moonlight and how that's maybe different than daylight? And then also, did you consider the life history of some of the different animals that you were having in that machine, given that they might have a sort of different behavior, but really fantastic talk.
Yeah, thanks, Paul. So I'll answer it maybe in that order. So we use typically red or IR LEDs to image the organisms. And the reason is exactly what you said, which is to have as low an impact as possible on their photo response, both in terms of their direct photo response to behavior, but also like photo response over longer scales, for example, that leads to diel migrations. So we do take care of that. So I think your second question was about the moonlight. So that's a very interesting question. So we haven't imaged organisms that have direct behaviors based on sun and moonlight yet, but that's definitely something that we are exploring, especially with things like coral larvae. So that's something that that's in the works. And so I think the life history and stages is actually quite an interesting question in and of itself. You would know based on the work we did in the embryology course that, we basically try to image them on as many days and as many parts of their developmental cycle as possible. But, the data is we are only getting started. I think what I showed is like really the tip of the iceberg. So for example, it would be fantastic to have an entire life history of an invertebrate all the way from an embryo to, more advanced larva. So that's something that, again, is something we are looking towards.
There's another question from Christina. Christina, go ahead.
Oh, sure. That was fantastic, Deepak. Thank you so much. My first question was what the most amazing thing you imaged in your PhD was, if you had to pick one. And then second question is, I was wondering what you think are the important advantages to cercariae using cycles of sinking and upward swimming to stay high in the water column, as opposed to like one upward gate that's perfectly tuned to counteract gravity.
Yeah, so that's a great question, actually. So maybe I'll start with the second one. So I think the answer is that we don't have a full answer to that, but I can speculate based on the kinds of data that we have so far. So there are two reasons. One is efficiency, right? So you are spending energy and you are energy limited. So it means that if you can somehow stay in a certain region of the water column, but spend say half the energy, you would think that you would select for that as a behavior. And the other reason is that if you only have an upward gate, you actually see something very interesting, which is that the larvae pretty much will congregate at the interface. So the cercariae will congregate at the interface. And that's actually a very interesting question, whether that actually happens in the field. We don't know. But you could, for example, think that that might open them up for predation. So another thing to remember is that, I kind of painted this very simple picture of, the snail and the human, and the cercariae goes from the snail to human. But there are like so many things predating on the cercaria. There are other types of cercaria, and there's so many. There's basically a whole ecosystem. So the behavioral selection, we need to think of that carefully, because it's not just for. Host seeking is probably up there, but there might be other pressures on the behavior as well.
Then there's a question from Lucas.
Oh, sorry, and I'll answer your first question too, which is I can't pick one. I think the diatom still ranks very high, just from like the shock value, because like I didn't expect to see what I saw.
Thanks.
Lucas?
Hey Deepak, thanks for everything. That was really inspiring and super amazing. I just have a question about, you studied the motivation for gravity machine as saying we want to understand, to link microscopic swimming behavior with larger sample that may be many organisms swimming together. Mm-hmm. Do you have a design idea of, say, slightly larger gravity machine that would enable you to actually maybe lose a bit of resolution at the microscopy scale, but say include more organisms and understand how their density, distribution evolves, etc.?
Yeah, actually, so Manu, Adam, and I have actually thought about this. It's something very interesting. So this applies both in terms of imaging larger organisms, but if you are, as you said, willing to lose a bit of resolution, so we have basically thought about a system where you don't track a single object, but you track the center of mass of a group of objects. So that's something that we are actually very excited about. So that would, I think, go towards doing the kinds of things that you're talking about, to see if, are there collective motions, what are the kinds of length scales that are involved in the collective motion. So remember that there is gravity always. So you need to be careful about defining collective motion in a system where there is like this global director field, but it'll be something very fun to image. Yeah, definitely. Thanks.
Maybe a last question, Julia, depending on time.
Yes,
so
I think one last question.
Last question, yeah.
Yes.
Buyang?
Yes. Hi, Deepak. Thank you for your talk. That's very inspiring and joyful. What were the challenges when imaging the planktons on site? I mean, why did you have to move your lab onto the ship or near the coast to image planktons there? Can you just bring the planktons into the lab and image there? What are the challenges associated with this?
Yeah. So yeah, thanks. That's a very relevant question. So the reason is that I think, so in some cases it is possible to bring plankton to the lab, but for many of the things that I showed you today, so these plankton are really part of a larger life cycle. So for example, all the invertebrate larvae data that I showed you, these plankton, they are, after fertilization, they develop through multiple larval stages. And we have actually successfully, grown them completely in the lab, done spawning in the lab and so on. But I think, first of all, going to Hopkins, for example, is just inspiring by itself, because you're in the natural environment there. And you get access not to a single organism, but multiple organisms. And you have the kind of infrastructure that you need to kind of keep these organisms happy, for lack of a better word, which is to say that you can grow them in the kind of conditions that work very well. So it's both access to organisms as well as kind of the infrastructure that's easier in these places. On the other extreme there are planktonic organisms that are currently not possible to culture in the lab. And they definitely decay if you try to bring them to the lab. So there, currently, we don't have any other option but to take the lab to the ocean. And that's kind of, I think our trip to Hawaii on Kilomana really showed us that, because there are many plankton that we imaged there that currently not many labs can actually keep in the lab. Yeah.
Thank you.
Well, perfect. I'm sure that it probably are other questions, but we have a fairly tight schedule. So I want to apologize if I have to basically end this part of your awesome presentation, Deepak. Thank you very much. And thank you to all the audience. And so we're going to close this call now. And Deepa, because we had a little bit tight with time, it would be great if by 11:05, in you know, slightly more than 5 minutes, in 5 minutes, we are going to, you know, your committee, and you are going to meet in this Zoom call.
Yeah.
So Deepak, which link are we using for the closed door?
So it's the private session link.
It's a different link.
I can send that again in case that's easier to find. Yeah.
That would be fantastic if you can remind your committee exactly what is that.
Yeah, exactly. I'll do that.
Perfect. Thank you very much. And thank you very much to all the audience. It's been fantastic, and looking forward to see you in less than 5 minutes, basically.
Yeah. Thanks everyone for joining.